right to it.
Yes. Okay. So I'll just start off again. So as I mentioned, I'm Ava, and together with Jesse and on behalf of the Bhatia Lab, I'm so excited to tell you more about our image, Making the Cut, probing enzyme activity with nanosensors. As you know, cancer is characterized by the uncontrolled growth of tumor cells that spread and invade the tissues in the body. But how are cancer cells actually able to do this and invade and spread in this way? It turns out that cancer cells produce a special type of protein, an enzyme that's called a protease, which I'm showing here as these blue Pac-Men. And what these proteases do is they actually can break down the tissue surrounding the cancer cells, allowing them to spread and invade. These protease enzymes are able to break down the tissue in this way because they have this very special property, in that they're actually able to recognize other proteins, which I'm showing here with this red curve, and cut them, breaking them down. In the Bhatia Lab, we're very interested in understanding more about what these proteases are doing and how they contribute to cancer. To do that, we used engineering methods to design nanosensors that can measure how and where these protease enzymes are active. The nanosensors we build are designed to be recognized and cut by proteases. And we've designed them in a special way such that a part of the sensor that's released by cutting can actually bind tissue at the precise spot where that cutting happened, such that we can visualize protease activity using microscopy. When we test these sensors on slices of tissue, like from a patient's tumor biopsy, we can visualize and localize where these cutting events happen in order to understand where these protease enzymes were active.
So Ava has introduced you a probe that allows us to visualize where these certain enzymes called proteases localize in a tissue. And so our ultimate goal was to apply them to cancer and to learn how proteases, the role that proteases play in cancer. But before we applied this approach to cancer, we first wanted to test it in a simpler and more well-defined tissue. So here I'm showing you a zoomed in section of the colon, which we sliced and mounted on a microscope slide. So each blue dot that you see here represents one cell nucleus, which you can see are all over the colon because all of the cells in the colon have a nucleus. The cells labeled in green are called epithelial cells, and these are the cells that make up the lining of the colon. These cells are known to produce large quantities of proteases, which are thought to help the body maintain a healthy relationship with the trillions and trillions of bacteria in the gut. So when we applied our protease activity nanosensors to this colon slice, we expected to see the green cells light up because these are the cells that come into contact with those gut bacteria, and this is exactly what we saw. So as you can see here, the magenta signal corresponding to protease activity is illuminating the green epithelial cells. So these findings were extremely exciting because they proved to us that our protease nanosensors can be used to visualize protease activity in a tissue, and suggested to us that we could maybe apply the very same approach to many different diseases, including cancer. So since we captured this image, we have done exactly that, which has allowed us to shed light on the many important roles that proteases play in multiple different malignancies, including prostate and lung cancer. And our lab is now working on using this information to target protease activity in order to enable earlier detection and treatment of cancer. So with that, we will just conclude and thank the people who made this work possible. In particular, our advisor, Dr. Sangeeta Bhatia, who has encouraged us to take risks and get creative with our science. And thank you also to the Koch Institute and our generous funders for supporting this work. And finally, thank you all for joining us here tonight.